Hello, good morning, good afternoon, good night, everyone. First of all, I want to thank the organizers for the opportunity to present this work in our symposium here. So I will discuss the effect of analytic choices on fMRI results and what we can do about it. And this describes a recent project just recently published in Nature. And so the first part, NARP's overview and context. So first of all, what is NARPS? The acronym, the Neuroimaging Analysis, Replication, and Prediction Study. You can read all about it in narps.info, all the details. And now in this introduction, in this first part, I'll give you the overview of the project. So basically the question is, what would happen if 70 independent groups of research teams analyze the same fMRI dataset? Sure, when we embarked on it almost 4 years ago, we didn't know it would be 70, but this is basically the underlying question. If all the research groups get the same data set, what will be the results? So today in our symposium, so first now I'm presenting the context and overview. I'll be followed by Rotem, who will present the findings. Remi, Remi Ga was one of the members of the analysis teams, will present his view as a team member. Jeanette Mumford, we'll talk a lot about the statistics, the nitty-gritty details, and a few takeaways. And then at the end, after the three of them, I will summarize and discuss what we believe are future directions after following this project. So the background that had led to this project is, first of all, the replication crisis. I'm sure you've heard about it. When researchers have tried to replicate findings in psychology, not all of them replicated. Less than half, and then economics, and then even cancer biology. So this is the starting point. And this is probably one of the reasons is the researchers' degrees of freedom that are when humans make research, we have choices we make along the process. And this was suggested to be part of the reasons why there is a replication crisis and not all studies replicate. Now, specifically in our field has been shown using simulations that there are multiple ways to analyze the same fMRI dataset in the pre-processing stage mainly, and also during the modeling stage. So we knew all about it. The approach we decided to take is the many analyst approach, previously applied on behavioral data, where the same data is used but analyzed by many groups. But it has never been done before in neuroimaging. And finally, I'll explain that we use prediction markets, that this is a tool that is commonly used to try and predict biases in a certain field. Briefly, participants receive a certain amount of money, and then they trade in a stock exchange on their beliefs. They trade this money between each other. It's commonly used many times to predict elections in Europe, etc., and we used it here, as you'll soon see. So the initial impetus for the project actually came from the economists, from this group of economists that has used prediction markets before in other fields. So Felix Holzmeister, Jürgen Hober, and Michael Kirchhoff from Innsbruck from Austria. Anna is also there, and also Anna and Magnus Johansson from Sweden, and Colin Kammerer from Caltech. Now, very COVID-19 appropriate. We have never met with them before. We've only met with Colin, and only recently I went to meet Felix. Everything was done remotely and online, all the discussions and deciding on the project. On the neuro part, it started actually without Rotem, who joined in later for the main part of it, but Russ and I were there working together with the economists on the project. So the aim was to test in the wild, as ecologically as possible, the variability of the analysis of fMRI data. So take the same data, and we told teams to analyze it as they would in their own research group, to test the variability really in the wild. And then we wanted to apply these prediction markets on the prediction. What do people think if the results will replicate or will not replicate? So the overview, first, almost 4 years ago, we started talking about the project. Then we collected the fMRI data set between 1 year here at Tel Aviv University, the Imaging Center, between July 2017, May 2018. And then for 3 months, or a little bit more than that, 70 analysis teams analyzed the data. When it ended. We ran the prediction markets, and then we wrote the manuscript and we sent it. So the task we decided to use, so I thought about it will be, so I study in my lab. Usually I do a single study, single analysis pipelines on value-based decision-making. And I thought this task was appropriate because it had been used before. I thought it's a relatively simple task for our field. And it's called the mixed gambles task, published initially 2007 and then followed by others. And very briefly in the task, participants are presented with prospects. First of all, they're endowed with a certain amount of money, for example, $20. And then they're presented with a series of prospects of gambles. Each gamble has a potential gain amount, potential loss amount, taken from a certain matrix of gains and losses. And the participant knows that they need to decide if they want to accept or reject the gamble. And at the end of the experiment here, accept or reject, at the end of the experiment, they know the computer will randomly draw one of the gambles. If they decided to accept, a virtual coin will be flipped. If they get the gain, they win the gain amount, or they can lose the loss amount. Now, this task has two main versions. One of them, equal and difference. This is following the work of Tversky and Kahneman that showed that losses loom larger than gains. Then this one has the gain amount is double that of the loss amount. Whereas in the equal range, it's the same range, 5 to 20, 5 to 20, versus 5 to 20 losses here in the equal and difference, and gains is 10 to 40. And there was a debate in the literature, what are the brain regions that have been activated by each of these tasks? So we decided to scan this task and ask the relevant questions on this task that had been done before, so people can go back to the studies published on these tasks. So Roni Ivanir, my master's student, she scanned 108 participants, 54 in each group, here at Tel Aviv University on our Prisma scanner with the multiband API, a 1 second TR, relatively high resolution. The data set is, of course, open and had been published. Okay, so this was the task, and then we decided the different hypotheses on the task. And now I'll hand off the Symposium to Rotem, who described what we did and the results of this analysis by the many teams. Thank you very much, Rotem, Remi, and Jeanette, for presenting all the results and the perspective and the statistical takeaways. Now, in this second part, my aim is to summarize and give the future directions, what we believe is the take-home message of this project. So first of all, as Rotem had presented, the results are what they are. It is what it is. If we look at the hypothesis and the way that we usually test them and see them in published papers in the field, there's large variability in the whole brain corrected results. It is what it is. Now, there's great agreement about the amygdala hypothesis. And great agreement, people usually didn't find them. And also maybe many people found the activations for the negative V and PFC. However, if you look at the other hypotheses, there's large variability in them. And it is what it is. Now, the nice thing about the way this study was set up, I think, is that one might say, there's a problem with the signal, et cetera, and V and PFC. Well, no, hypothesis number 1, number 5 are on the same region in the brain. So this serves as an internal control, and it's not anything about the way that people had done or analyzed the data. Now, I'm not saying one hypothesis is better than the other, but these are the results. And this is what we found. Now, I think this result was surprising, at least to me. So when Rotem and I saw the binary results in the Google form everyone filled in, then we saw there's large variability. But it took some time until we analyzed the unthresholded maps, that we saw this large red cluster that they actually uncorrected. The intermediate statistics, actually, they are pretty correlated with each other. So while there is this red and green here, the large variance, this is for hypothesis 1, large variance in reporting of whole brain corrected results, then there is much more correlation in the intermediate statistics. And this is, I think, very encouraging for a field to know that the underlying data are pretty similar to each other. Now, as you've seen in Jeanette's talk, the sleuthing that she has done on this anti-correlated cluster, and I think we said this is in the wild. This study is trying to look for what is ecologically valid. So people make mistakes all the time. We all make mistakes. It is possible that here the the mistakes, maybe because this is a part of a large project, that when you just send one result to a paper, you look much more closely. But I think people make mistakes. We know that we sometimes find them. So again, this is another ecological aspect of research. Now, the markets, the bias that was shown in the markets, again, lining them up based on how many people confirmed a certain hypothesis. So these are the amygdala ones, and this is here the negative VMPFC. If we look both at the team members that had inside information and the non-team members, we have a bias in the way people think. They're optimistically biased. And this is probably due to the publication bias. So for my field, when people would ask me, okay, would you get a positive VMPFC? Of course I'll get for gains a positive VMPFC. So even people from the team members who didn't find it, they see here, well, I'm sure the others got it. And this is due to the bias that mainly significant results in recent years, mainly significant results were the ones that were accepted for publication. So this is what we think is usually the case, but I had a few people that made a lot of money, experts from the field, that said, well, Tom, we all know that we don't always get VMPFC. So they said, thank you, we could make money in the prediction markets. So we see a marked over-optimism by both the team members, they're really shifted by a constant, and the experts from the field, the non-team members. So this is the publication bias being expressed, I think, in the over-optimism bias. Now, we saw in Remi's slide about pre-registration. So I think this is a very important take-home message. So mostly what we did was we analyzed things in many, many ways, and then we reported one of the ways, why we weren't aware to the implications of this. This was not done on purpose. Pre-registration, what it allows us to do is to hypothesize initially, and in my lab, this is what we've been doing for several years, is that we pre-register the analysis pipeline that we'll do, and then this is what we report. Of course, we can still report all the exploratory analysis we want in the world. That's perfectly fine. Everybody talking to me about pre-registration, we can still do any exploratory new results we want, but we have a greater confidence in what was pre-registered compared to what was reported. Now, finally, what we really support here is the multiverse analysis. Is why don't we, or if we can, analyze all the different ways and report all the different analysis ways. And then the way I envisioned it is having another multiverse statistical section, is that to report, okay, so these are the results that are the mostly robust, and they replicate, and they're there in most of the analysis. Now, I think prior to NARS, what people have mainly thought about multiverse, about thinking about the thousands and thousands of ways to analyze the data. That if you saw in the results, because we shared both with fMRI prep or without fMRI prep, I mean, after pre-processing or without, there was no difference for our study. So therefore, right now in the lab, what we're working is on a way, like on a small scale multiverse analysis, a single lab multiverse analysis that will actually play more with the way that we do the actual modeling that influences the result, because NARPS is a multiverse with 70 teams. So why can't we do this? But until it's there, I suggest we all pre-register. I think that's a very important take-home message. So to conclude, what we found, a lot of flexibility in analysis. It influences reported results. These are the whole brain corrected results. The unthresholded maps, they're much more similar. This was a surprise, at least to me, than the final result. And what we did, we analyzed the variants that had led to the sleep. It's not only the final stage, not only area definition, but of course, as more as we strip the variance, then we get to the final, to a closer outcome. Less variance, closer outcome. People are optimistically biased in the field, probably due to publication bias. And as you've seen how important it is, we could have not gotten to the conclusions of what is similar, what is dissimilar, if we didn't have the raw data, the uncorrected statistical maps. So share them with every publication, share the code, share analysis code, and pre-register. This will really help until we have better statistics in place, and the multiverse approach, which we believe is really the best one that is out there to solve this. So the conclusions is, first of all, it's very important. I think part of the success here, there was no incentive to p-hack because there was no ground truth. And it's also, the publication wasn't dependent if you found significant results or not. So people really reported what they found. And of course, we encourage others to try. What we have here is a case of n equals one task. This is the task we tested. There are many, many, many other fMRI tests on Open Neuro. Try them out. And finally, our conclusions apply, we believe, to every empirical research, biology, chemistry, everything that involves humans that make decisions. And these bifurcations, and my friends from biology, chemistry tell me, wow, Tom, you're really brave to have done it in your field. We're surely it will be the case in our field as well. So, and hopefully people are starting to discuss and follow this. So I want to thank each and every one of the co-authors on this paper, all 197 on them. You know, Dr. Rotem, Botvinik-Nezer, the first author, was a PhD student, now postdoc with Tor Wager. All the rest, the senior co-authors, Tom Nichols and Ross Poldrack. And of course, the funding agencies that allow to scan this, Austrian Science Foundation, Israel one, Diane Wallander and Tom Idilus Foundation. And thank you very much.
Hello, everyone. I'm Rotem Botvinik-Nezer, and I was part of the team leading NARC as a PhD student at Tom Schonberg's lab at Tel Aviv University. And I'm a postdoc at Dartmouth College in Tor Wager's lab. And I will continue from the point Tom stopped, his introduction talk, and we'll focus on the analysis part and on our findings. So as Tom described, our first and main aim was to estimate how similar are the conclusions of different researchers when testing the same hypothesis with the same data. And for this aim, we recruited 70 independent group of researchers from all over the world to participate in the analysis of our study of NARCs. Overall, there were 180 members, up to 3 per team. They came from many different countries and from different positions, and they were all offered co-authorship on the paper in return for their participation. They had up to 100 days to complete and report the results from November 2018 to March 2019. And the data set that Tom described was collected for this project. It included 108 participants, performing each of them performs one of the 2 versions of the mixed Campbell's test. And the data set was shared with the analysis teams, both the raw data set and an optional version of the data set pre-processed with fMRI prep. The data set was shared on Open Neuro. It's publicly shared, and it was also published as a data descriptor, so we can all access it if you'd like. The teams received access to the data. And as I told you before, we wanted to do it in the wild, so to see really in an ecological manner our things in the actual literature. So we minimized the instructions, and we asked them to freely analyze the data using their standard analysis techniques as they would for a paper in order to test the following 9 ex-ante hypotheses. The hypotheses were pretty fine a priori based on a debate and the published literature of decision-making under risk. And each of them consisted of a specific activity, either a positive or a negative activity in a specific brain region related to one component of the task, either the potential gain or the potential loss amount, and one of the experimental groups, either the equal indifference group or the equal range group. And for example, hypothesis 1 was about increased activity in the ventromedial prefrontal cortex as potential gains increase for the equal indifference group. So this is parametric modulation, and it is a representative hypothesis out of this 9 hypotheses. So the teams were instructed to independently test those 9 hypotheses with the data sets we share with them, and then to report a binary decision, yes or no, significant or not significant, for each hypothesis based on all brain corrected analysis. In addition, we asked them to report in details their methods. We followed the COBITUS guidelines and created a form with 11 pages where they had to report each step of their analysis. And we also asked them to create a new collection on NeuroVault and upload 1 thresholded and 1 unthresholded statistical map for each hypothesis on which they base their decisions. So now let's go back to the key question, right? What would happen if 70 teams analyze the same data to test the same hypothesis? So here's how it looks like. First, the high analytical flexibility in our field was demonstrated by the fact that no 2 teams chose the exact same analysis pipeline. This was amazing. And the teams made different analytical choices at different stages of the analysis. For example, which software they use, which pre-processing steps they included, and with which parameters, how they modeled their hypotheses, which regressors, which contrast, which thresholding methods and correction methods for multiple comparisons, how they defined the region, and how they decided whether there was a significant activity in this region or not, and many other analytical choices. So 70 teams use 17 different methods, but did their results differ based on this? So the short answer is yes. We found substantial variability in the results across teams, but let's look at it in more details. So here you can see a plot of the results. At the y-axis, you can see the hypothesis number, and on the x-axis, you can see the proportion of teams reporting a significant result. I want to remind you that the maximal disagreement is 50% of teams reporting a significant result. And this is if the results were random, we would have been expecting this, right? Half of the results to be significant and half not significant. 0% means maximal agreement on a non-significant result, and 100% means maximal agreement on a significant result. So let's start with hypothesis 5. 84% of the teams reported a significant result for this hypothesis, and overall there was agreement it was significantly supported by the data. This was about negative activity in the VMPFC as losses increase. Hypothesis 7, 8, and 9 about the amygdala, there was an agreement about it not being significant. Only 6% of teams reported significant results for each of those hypotheses. Interesting, by the way, not the exact same teams across those three hypotheses. And for the 5 remaining hypotheses, 1, 2, 3, 4, and 6, the percent of teams reporting significant results varied between 20% to 40%, which is about midway between complete random and complete agreement. I want to remind you again, those are the same teams, the 70 teams testing the same data to test the same hypothesis. So then we ask ourselves, okay, so what are the sources of this variability? Russ Poldrak led the analysis part, and you can see here URLs to the fully reproducible version of the code used in our analysis with a Docker container. And first of all, we tested what parameters of the analysis pipelines were related to more or less significant results. We performed a logistic regression with some of the analytical choices included in the model, and reporting a significant or reporting a non-significant result as the outcome variable. So we found that the estimated smoothness of the unthresholded maps, which was estimated from the unthresholded maps with FSL's smoothest. So it's not only the applied smoothing parameter, but other pre-processing steps also affect this value. And it was significantly related to reporting more significant results. Also using FSL rather than SPM and using parametric rather than non-parametric thresholding methods were both related to more significant results. Users who are not using fMRI prep pre-processed data was not related to more or less significant results. Neither was modeling or not modeling the head movement. But I want to emphasize that there were many other analytical choices that we couldn't include in our model. For example, how the teams defined the specific regions, the VMPFC, the ventral striatum, or the amygdala, which regressors they put in their model, etc. Many other steps that we couldn't model because although we had 70 teams, there were too many different options, different methods they used, and we didn't have enough power to test those. We also ran a non-parametric bootstrap analysis to verify those findings, and it provided support for the estimated smoothness result, but inconsistent results for the other parameters. The next step was to look at the level of thresholded maps. Is the variability also present at this level, the thresholded maps? So you can see here the overlap across teams for hypotheses 1 and 3. Those hypotheses share the same statistical map, all-band statistical map, because they are for the same contrast, only different brain regions. And you can see voxels that were active in the thresholded maps of at least 10% of the teams in color. Green is less, red is more, and the color represents the proportion of teams where this voxel was active. And you can see that the agreement was largely limited to which voxels were not activated, most of the voxels. And when looking at the number of active voxels or their location, we saw considerable variation across teams. For example, the number of activated voxels varied from 0 to tens of thousands across teams in each hypothesis. Then we proceeded to look at the level of unthresholded maps. And surprisingly, at this level, we found relatively high correlations between the maps of most teams. So here you can see a correlation matrix with spiral correlation between each pair of teams. And it was clustered to 3 clusters with word clustering on Euclidean distances. And so you can see in red the main cluster of most teams that had really relatively high correlations between their maps. But nonetheless, if you look at their binary results, you can still see at the top in red, it means they reported a non-significant result, and in green, it means they reported a significant result. So even 50 teams in this cluster, they were highly correlated in the unthreshold level, but they still uh, um, reported very different binary results. Another thing you can see here is the blue cluster with 7 teams that the results were anti-correlated with the other teams, with the main cluster. And there we found some modeling issues, but Jeanette Mumford will elaborate more on this later in this symposium. Here you can see the same metrics for the other hypotheses. Overall, across hypotheses, there was a large cluster of teams with highly correlated maps, but nonetheless different reported final decisions. So then we said, okay, so we have correlated unthreshold maps, but they led to very different binary results. So it suggests that the thresholding and region specifications, less stages of analysis from the unthreshold maps to the binary results, are the main sources of variability. And we wanted to test this. So we took the unthresholded maps of the teams and we applied 2 different consistent thresholding methods and a consistent ROI specification on those maps to see if we get different results. And indeed, the results were different, and it's more complex than this. But if I would say it shortly, then qualitatively, we got a similar variability in results, even when we did this. So we reduced the variability by making using a consistent method for both thresholding and ROI specification, but still we got very different binary results. So you can't say that the variability comes from only those analysis steps. Finally, we performed an image-based meta-analysis or consensus analysis and on the unthresholded maps of the teams, and we found significant active boxes for all but 1 hypothesis and confirmatory evidence for 4 of the 9 hypotheses. Overall, this meta-analysis on the unthresholded maps revealed convergence across teams and seems like a promising future directions to use in different pipelines, and Tom will elaborate on this more later in this symposium. And now let's go to the second aim, as Tom described. Our second aim was to test whether researchers in the field could predict the results, that is, to predict replicability and reproducibility of fMRI findings. And to do so, we used prediction markers. They were also used in previous replication studies in psychology and in economics and more. And in such scientific prediction markets, researchers trade on the predicted outcomes of scientific studies. And here, for example, they traded on the predicted proportion of teams reporting a significant result for each hypothesis. Later, participants are paid based on their performance in the markets. So we ran 2 different prediction markets. The first one was for researchers from the field who did not participate in the analysis part, and 65 active traders participated in this prediction market. The second one was for analysis team members. 83 of them participated. And importantly, the markets were run after all teams submitted the result, but the results were kept confidential. So during the prediction market, analysis team members knew only their own results, but not the results of the other teams. Here are the results of the prediction market. In pink, you can see the actual fraction of teams reporting a significant result for each hypothesis. In green, you can see the final average predictions of the non-team members prediction market. And in blue, the final average predictions of the team members prediction market. And as you can see, it's clear from this figure that almost all predictions were over-optimistic. So both markets, participants predicted higher rates of significant findings across teams. And the amazing thing is that even team members who themselves saw the data and rejected many of the hypotheses, they still predicted higher rates of replicability. Another interesting finding was that traders in the team member markets were highly calibrated, meaning they consistently predicted high proportions, but their predictions were highly correlated to the actual results. So to summarize this exciting project, we gave 70 independent teams of researchers from around the world the same fMRI dataset, and we asked them to test the same 9 hypotheses. And we found that there was some substantial variability in the analysis pipelines they used, leading to substantial variability in the binary reported results, although the unthresholded maps were relatively correlated across most of the teams. And we also found convergence across teams in an image-based meta-analysis, and an optimistic bias in the field regarding the replicability and reproducibility of results. And I want to thank all 196 co-authors who made this happen, and particularly to the analysis team's members, to the participants in the prediction markets, and to Tom Schonberg, Russ Baldwin, and the entire NAPS team, as well as the main funding agency who enabled this project. And after me, Remi will present this project from the eyes of a member in the analysis team, followed by Jeanette Mumford, and then again Tom Schonberg. Thank you for listening.
Hello, everyone. My name is Remi, and I'm a postdoctorate at the Cross-Model Perception and Plasticity Lab at the Université Catholique de Louvain in Belgium. And I was one of the persons who analyzed the data of the NARPS project. And I'm going to give you sort of the insider's view of what it felt to participate in this project. And in preparing for this symposium, I also interviewed and talked with some other scientists who participated in the project, and then ran a small survey among the analysis team to sort of get a more of a collective point of view of how it felt. And I'm going to use some of that information to sort of complement and contrast my own point of view on all this. Okay. So here's the outline what I want to tell you about. First, I would like to tell you about the differences on this type of project, large collaborative project, compared to the one team, one data set that we're more familiar with. Then I want to tell you about how using data standards and pipeline standards can really help speed up your work. And then I will conclude with some of the lessons that we have learned throughout this project. So first of all, differences between this type of large scale, many teams project and like the one team, one data set approach. Well, first of all, you do have obviously a diversity of motivations of why people might want to join this project. As Tom explained before, there is the issue of reproducibility and replicability that is present in neuroimaging and in psychology, and there's been a lot of discussion in the community about this. And this was clearly sort of my main reason for joining this project. But clearly some people that I talked with came more from the decision-making angle and really sort of were more interested in the fact that this was a decision-making study. And if it had not been about decision-making, they're not sure they would have joined the project. Some other people mentioned that this was a very good opportunity for them to try new tools, to get their hands on a BITS data set, which was the way the data was structured for this project. Or to sort of implement some more Python thing in their pipeline. Whereas some other people, on the contrary, wanted to sort of stick to their very typical pipeline and evaluate how it fared compared to how other people analyze the data and other pipelines might have used. Many people also mentioned that this was a very good training ground. That, for example, you have a new PhD student who has joined your lab, and this gives you an opportunity to pair that person with a postdoc who's more experienced and who can sort of help them and give them through their like first hands-on experience when it comes to analyzing fMRI data. About 2/3 of the people also that I surveyed also mentioned that definitely knowing that there were going to be an author on the paper was affected their decision to participate. And finally, quite a few people also mentioned that they could not acquire data at the time when this project was happening. So it was a perfect opportunity for them to sort of keep on working on some data. And I think that that last point sort of highlights the importance of sharing data in general. Next thing I want to sort of tell you about the diversity of expertise that you have when you're in such a project. Well, first of all, for example, the expertise in terms of decision-making science. You clearly see sort of this bimodal distribution where 1/4 of the people mentioned that they had already run a decision-making study before. And the rest of the people like spread from like having some experience decision-making into decision-making or no experience at all. And the red square just here highlights where me and my team members sort of stood on that issue. We clearly had no prior experience into decision-making. And then finally, I want to also briefly talk about the incentives that motivate people to join this project or that push them to sort of keep on working on through the project. And one of the big differences that clearly statistical significance was not the goal of the project. The project, as some people said, just promoted thorough and honest analysis rather than chasing significant blobs. And some other people mentioned just authorship was completely unrelated to whether or not our reported results endorsed a hypothesis. So in other words, all the incentives to p-hack were just completely gone. And I would like also to stress that, especially when you have no prior experience into decision-making, you have no particular emotional attachments to the results. So, the results are the results and, this is it. But some people also mention that, yeah, but it's also meant that all the incentives to spend extra time reviewing the decision-making literature were also not present. So this might lead you to think that some people maybe just rushed through the analysis and were not very careful. But actually remember that all the results from each team would be compared to those of the other teams. So I actually asked people, did you feel extra pressure to sort of get this right because you didn't want to stand out in terms of results compared to the other teams? And clearly, in general, people felt that there wasn't this extra pressure that this would be a team thing, but their results would be compared to those of others. And they really went through, quite a few people mentioned that, yes, they went through extra rounds of checking their code, making sure everything was correct. And I definitely felt that extra pressure in this project. And then I want just to talk about the fact that getting help on such a project is a bit different from what it is on your typical type of analysis. For example, remember that each team had to analyze the data independently. The idea was that we didn't want to have any sort of information leak between the teams. So we couldn't go to our usual forums and ask very specific questions about our data set. So I guess what I'm trying to say in a way is that, well, the first rule of the NARPS Club is you don't talk about the NARPS Club. And through the whole thing, we couldn't sort of tell our friends what we were working on. But, if you spend some time on some forums, like on Twitter, like I do, and some of your friends are, posting about high temporal resolution fMRI and parametric regressors, which were some of the characteristics of the data set and the experimental design, then, you sort of manage to figure out what your friends are actually working on these days. And in the end, you sort of figure out that they're also working on the NARPS project. So then I want to tell you about how having standards and using standardized data set and pipeline can really help speed up your work. This is important because remember, we only had 3 months to analyze a data set with 100 participants. And for some of us, we had no prior experience in terms of decision making, analysis, and the type of experimental design that are involved there. So one way things could be really sped up is that the data were shared as a bits data structure. So it's the brain imaging data structure that I tend to usually refer as the Marie Kondo of neuroimaging data curation. So instead of dumping a whole bunch of DICOM files on people with just irregular file names, you have this structured folder hierarchy with very regular file names. And the advantage of this type of data structure is, well, there are several of them. First of all, well, you only need to figure out how things are organized just once. The idea is that if you have read the specifications, you pretty much know where to find every bit of data and metadata in this data structure. So you know where are all the characteristics that you need to sort of analyze the data in this or that other way, in which file to look it up. And this also means that once you have seen one big data set, you pretty much have seen them all because they're very, very similar in terms of how they're organized. So you do, it's a good way to sort of gain a lot of time in analyzing the data. The other thing is that this base data sets can facilitate automation. So you can clearly, because a software will also know where to look for the data and the metadata, you can clearly create this pre-processing or quality control pipeline that will just run on their own if you just tell them it's a bits data set. And that's what the NARPS team did actually. They just run the analysis, the data through the fMRI prep, which is a pre-processing pipeline. And they also use the MRI QC pipeline, which is a quality control pipeline, and then gave all of those results to all of the teams. So this was really a way for us to save a lot of time, and also a good way for us to get full reports on the quality of the data and know if some subject had to be maybe kicked out, or some time points for a given subject had to be kicked out because there were too much movement, for example. So this really sped up just getting a big overview of all the data, and also to sort of finally just get directly to the analysis and skip all the pre-processing, for example. And finally, I want to tell you about pre-registration. We were one of the few teams who pre-registered data analysis. So usually when you analyze your data, you have to make a lot of analytical decisions, right? So you might just go one certain analytical path. The results in the end maybe don't make quite a lot of sense. So you go through another analytical path. And you do that until you have some result that actually makes sense. And these are the results you report. Well, pre-registration is the idea that you decide in advance of all the analytical decision you're going to have to make. You run that single analytical path, and that's what you report. Obviously, this is most appropriate for replications because you know in advance a lot of the decisions that you might have to take because someone has made them before. So that pretty much just allows you to front load all the decisional process. You do everything in advance. But that also forces you to make all of those decisions very explicit and not sort of to make them after the fact once you have seen the results. The other good advantage is then you can just create your whole pipeline in advance and there is no back and forth between seeing the results, changing the pipeline, and so on and so forth. So in the end, you do save a lot of time just not exploring the results. And finally, I think there's also a very important pedagogical advantage for pre-registration, and it can clearly avoid graduate students sort of tumbling in the dark, not knowing where they're going, and you can sort of create flow charts and even contingency plans to help you like structure how you analyze your data. And then now I just want to quickly go through some of the lessons that we have learned through this project. Well, first of all, I would just like to mention the fact that such large-scale collaborative projects are fun and are enjoyable, and more than half of the people said absolutely they really loved participating in this project, and that 64% of them would clearly do it again. So I think there's a market for more analysis and projects like this one. And then finally, I want to conclude saying that this has taught me that neuroimaging analysis can be very straightforward, that it can be done in a very short time, but that this also relies on the importance of sharing data and using just common standardized pipeline. And finally, I think one lesson that was made very clear to a few of us who pre-registered is that pre-registration for an fMRI study is tough. There's a lot of decisions, there's a lot of things you need to think about, and there's clearly a need in the community to create templates to help people pre-register the analysis and guide them through this process. And on that note, so here are the links to the survey questions and the answers that I got from the other team members. They're all on the Open Science Framework, openly accessible. And I would like to thank Olivier and Marco, who were my team members. I would also give a special shout out to all the people who took the time to be interviewed by me and answered all my questions about how it was to analyze the data and participate in the project, and then helped me sort of create this survey that I could circulate to the other teams. And finally, I would like to thank the whole NARPS team for organizing this and all the 69 other teams for making this fantastic project possible. So thank you very much for listening. And now I think that Jeanette will be telling you about some of the statistics of analyzing all the results of this project. Thank you very much.
Hello, so my name is Jeanette Mumford, and I'm from the University of Wisconsin-Madison. So the portion of the NARPS project that I'll be talking about has to do with figuring out some of the mysterious findings. And we were able to identify these problems because the uncorrected maps were shared and the design matrix information was shared. So I'm going to be focusing on that specific problem and giving some tips, not just for future neuroimagers, but for current neuroimagers. So often with studies, we focus on two things when we're first taking a broad look at the study. The hypothesis, so perhaps one day somebody was talking to their dog and they're like, man, I really feel like you understand me. I'm going to figure this out using the fMRI. So then they run some study and the results show some blobs on the brain indicating differences in the brain activation for the dogs when listening to pseudo words versus words. So on the surface, this is very interesting, but to properly really understand and kind of believe this study, we need to take a deeper look at the methods. So in this case, the methods section was really great. The study was really solid, which I could determine because the methods section was written so well. So we have a nice example of what the study paradigm looked like. The pre-processing details are very clear, and the linear model information was very clear. So what this allows me to then do is make sure that the questions that were asked in the hypotheses that were outlined in the introduction to the paper are actually the same questions that the statistical models were answering that were shown in the results. So I would like to focus on these two important aspects of what your method section should achieve. First of all, is whether it is a solid link between the questions asked and the questions that the model answers. And also that the description is clear enough that we could recreate the analyses on our own. So often that's thought of in terms of replication, but it's actually also really important in making that connection between the questions that were asked and the results. So there's a lot more to it than replication. So specifically, for the first point, I want to focus on whether the psychological interpretation of regressors matches statistical interpretation. This is something that I personally spend a lot of time helping people to do because there's often a lot of work involved linking these two questions. Also, if there is a mystery to be discovered in a study, is the information supplied enough to both see that mystery and dig deeper into it? And we'll see that in the case of the NARPS mystery, we did have that information. So this is my favorite mystery from NARPS, and you have seen this image in the earlier talks of this session. So what it is a similarity matrix comparing the unthresholded statistics maps, in this case, for the positive gains effect hypothesis, which was the first. And you can see that most of these were positively correlated with each other. But what's really interesting is this little blue band here. How the heck did some folks have statistics maps that were actually negatively correlated with the bulk of the other statistics maps? So that's really interesting. So I'm just going to be focusing on one of those specific examples, basically one of those blue columns. And thanks to having the statistics maps, we were able to identify that there was a problem. But generally, if it's a field that you're familiar with and you see a result that seems counterintuitive, it went in the wrong direction, it should trigger a little alert and cause you to look more deeply. Also, thanks to having really clear descriptions of what went into the design matrix, and in some cases actually having the design matrix helps to dig into this problem and figure out exactly what happened. So just a quick reminder about what this task was. So for each trial, there was both a gain and a loss amount, and then the subjects could accept or reject the gamble. There were two different setups. There was the equal indifference and the equal range, and you can see it was just differences in the ranges for gains and losses between those two data sets. So the specific hypothesis this was reflecting was the positive parametric effect of gains for the equal indifference group. So what that means is the positive relationship between gains. So as gains increases, does the bold activation also increase? So the issue here, again, the explanation of the negative correlation with the other statistics maps is an example of when regressor construction leads to confusing statistics. Specifically, I would say this was a case of a mismatch between something that psychologically seemed very logical and something that statistically was actually quite different. So although most of the teams chose to look at gains and losses separately, in this case, the focus was on gains and instead of losses, expected value, which you can see is gain minus loss divided by 2. So I'm simplifying the model here and kind of stepping away from the convolution and parametric modulation just to make it easier to look at it. So you could just think of this as the bold activation for a trial, and the way that's modeled as having some mean beta naught, and then the gain value, and then the expected value as well, which again is gain minus loss divided by 2. So looking at this parameterization of the model, your gut is probably saying, oh, beta 1, that's the gain effect. Well, if we take a closer look at the model and actually plug in the meaning for expected value, we'll see that that's not the case. So expected value is gain minus loss divided by 2. Now I'm going to combine like terms, and you'll see that the actual gain effect is not beta 1, but beta 1 plus beta 2 divided by 2. So that means in the case of teams that did similar model setups to this one, that gain effect was actually missing part of its parameter estimate. In this case, it would be off by a factor of beta 2 divided by 2, which is what caused that negative relationship. So the takeaway from this, well, first off, was the model wrong or not meaningful? It's really hard for me to say. I'm not a psychologist. Maybe there is a psychological interpretation, but what's important is the interpretations are certainly very, very different. It's not just the gain effect as was reported in the other models, but it's this gain adjusted for expected value, and whether or not that has a meaningful interpretation is to be determined. So that's the first takeaway. The second is that generally, just be careful if you're setting up a model where you have variables that make up different regressors within the model. That's usually where these troubles pop up. Another example where this can happen is if you have 2 time points and you're modeling time difference. And so you have time point 2 minus time point 1 as one regressor, and then you adjust for the baseline time 1. That can actually inflate type 1 errors. So what should you do? So I'm going to view this from the perspective of reviewing a paper as well as writing a paper, specifically the methods section. Kind of keep a list of things to check for. And a lot of these things that you're going to use to build up your list are actually going to come from your own mistakes. And if we don't learn from our mistakes, then what's the point of making them? So, and then there are other things that will go in there that are mistakes you'll see other people make. So these are just a few of my examples. Whether mean centering was done, it's often not necessary, and if done incorrectly, it can lead to really misleading results. So if it was done, I usually take a closer look at that. Also, orthogonalization is a bit of a peeve of mine because it's very easy to misinterpret results when orthogonalization is used. And I find that often the interpretations people give to orthogonalized regressors are incorrect. So I do have a paper on that, which I've listed here. And also certain software packages, so older versions of SPM actually does orthogonalization without telling you, so you can read the details of that here. Also, there could be a pre-processing step that you've personally seen makes a huge impact that was left out. So you could think about that, and so on and so forth. So the list is actually bigger than that. And perhaps we could generate a list as a community. A lot of these ideas are already incorporated into the COBITA's guidelines, but I think you could get even more refined than that. So when writing your own methods section, so I'm often told, you know what, I'll ask for something in a paper, and then I'm told, oh, there's not enough room. There's always enough room in your paper. You always have the supplement where you can dump tons of information. But I do think it's important for the information specifically about the regressors and the contrast to be in the actual manuscript. So this doesn't really take a ton of space. And I'm not talking about including the actual timing specifically, but at least having the description of the 3 pieces of information that go into each regressor, which are the onset time, the duration, and the parametric modulation if used. Obviously, if you don't have parametrically modulated regressors, you only have to explain the onset times and the duration. So again, you don't have to list out the times, but often you will have that diagram of your study paradigm. Just make sure that's very clear and you can easily refer back to that diagram to describe the timings of your trials and the durations of your trials. It's also helpful to explain how many trials are represented by each regressor. If you have a bunch of trials and they're divided into groups, just say, oh, there were 30 positive trials in this regressor, 30 negative trials, or on average, there were 15 correct trials and 20 incorrect trials or something like that, just to give the reader, just to help solidify the picture of the design matrix that the reader is forming. Whether or not derivatives were used, if orthogonalization was used, depending on the software used, what option for orthogonalization was used. So I definitely find that putting all this information directly in the paper, it's 100% possible, and it doesn't break the flow of the reader trying to piece together the whole story. There are also tools. So there's the NIDM results tool, the paper here sharing brain mapping statistical results with the neuroimaging data model. So what this tool does is it puts together all the information from your analysis. And although it was designed, I believe, with group level models in mind, you could load in a level 1 model, and then the design matrix for the level 1 model will be included. But do be careful. Convolved regressors are often hard to figure out exactly what they are, especially with parametric modulation. They're still useful, but it's nice to have those as well as the timing information. So generally, teleporters are really cool, but we shouldn't let our papers behave like that, where the hypotheses just magically transition to results. Instead, we want to make sure that our methods section is a very solid bridge that connects these two things and creates a complete story for the reader. It's also really important, you'll find, if you're trying to find, I was just working with somebody who's trying to find a task to use in her study. So she was interested in eliciting response in a certain part of the brain, and also, a task that generally did a specific function. And she kept finding them and she was very excited, but she was focusing on the hypothesis and the results. And then I would dig into the methods to see if we could use it. And I'd have to say, I can't guarantee that we can recreate this. So it's very frustrating for us. She did eventually find something though. So that's a wrap. I just want to say big thanks to those who involved me in this project. So Rotem, Tom, and Russ, and others. It was really fun to work on this specific problem, as well as see the progress of it. And a big thanks to all the participants who analyzed the data.